Great. So I'm very, very happy to welcome you all to the 8th month of the CGC monthly webinar series. So thanks to all of you who've been attending the webinar in these past several months. We've been very successful in continuing to host this exciting webinar series and showcasing the excellent work done by several of our users and our collaborative project awardees. So just a few logistical items, the resources from this webinar, the slides and the recordings and upcoming webinar info will be made available in the CGC website. You can visit the cancergenomicscloud.org/webinars. And as you all may be familiar with, these webinars happen on the 4th Wednesday of each month at 2 p.m. Eastern. So we have a couple more months in this year before we go on a short winter break. So stay tuned for the next 2 months as well. As always, please use the chat box for asking questions. At the end of the presentation, we will have a Q&A session. And we will post a link to a feedback survey. And it's very, very valuable to receive feedback from all of you so that we can cover more topics in the upcoming sessions. So with that, it's my pleasure to introduce our speaker for today, who is also one of the CGC Collaborative Project Award recipients, Ms. Margaret Linian, who is here with us today to share her experiences in using the Cancer Genomics Cloud to detect and rank mRNA dysregulated pathways using this tool called POTRA. She will also be highlighting one of the most exciting and new studies from their group. It's called the Polytherapy AI, which is a multi-institutional, multinational collaboration. And it's going to be developed as an interactive artificial intelligence platform for multiomics combination drug treatments. Margaret is a bioinformatics consultant who is currently collaborating with Dr. Valentin Dinu, who is a principal investigator at the Translational Genomics Lab, College of Health Solutions at Arizona State University. And she also collaborates with several other academia and industry people on the Polytherapy AI project. She has previously worked in academia as a computational research scientist and consultant at the Icahn School of Medicine at Mount Sinai, and in the biotech industry as a bioinformatics deep learning scientist. She has also worked as a bioinformatics researcher in the Department of Health Sciences Research at the Mayo Clinic, where she successfully executed a lab scale pathway analysis and graph networks project. So over to you, Margaret.
Okay, thank you so much for that great introduction, Sai. So I want to start by discussing the first 2 pan-cancer studies, and then I will get into the Polytherapy AI study, which is a very exciting new study I'm working on. Let's see. Okay, so first a disclaimer. This presentation contains information related to 3 experimental research studies by the authors and are not intended to guide clinical applications. The information presented here are the intellectual property of the authors and only represent experimental methods and results. External parties are prohibited from using any of the study-related tables, graphics, and results in this presentation without the permission of the authors. And the 3rd study, methods and results are already part of a manuscript in preparation. So our research studies focused on messenger ribonucleic acids, known as mRNAs, and epitranscriptomics, because mRNAs are important molecular targets of disease because they can dysregulate hundreds of protein genes. mRNAs are also part of the stress response at the translational level and are mediators of carcinogenesis. mRNAs are especially important in our lifetimes due to their ability to be leveraged in vaccines and medicines. And according to Moderna, mRNA medicines make up a new category of medicines that are sets of instructions that direct cells to make proteins to prevent or fight disease. Our work also focuses on exploring patterns in methylated ncRNAs because it is considered a novel process in the arena of epitranscriptomics and is thought to contribute to several cellular functions in human disease, from regulation of post-transcriptional gene expression to adaptive cellular responses. And in cancer, non-coding biomarkers have also been shown to significantly dysregulate the transcriptome. So here's an overview of the translational research studies. That includes our CGC TCGA data browser methodology here, where you can kind of see it. We're basically importing primary tumor samples and solid tissue normal samples based on several criteria. And so we also have here workflows and results from all 3 studies. The images here highlights the importance of using epidemiological approaches such as stratification to achieve a better understanding of the data set, so, which is something you can always get through PCA and clustering. And this helps with understanding our conclusions, of course. And our tools included Rabix, Python, R, Docker, POTRA, Neo4j, GitHub, and the Cancer Genomics Cloud. So the first study is called the Pan-Cancer Exploration of mRNA-Mediated Dysregulated Pathways in the Cancer Genomics Cloud. It's also, the manuscript is actually available on bioRxiv for anybody who wants to read more about it. So in the CGC, the bioconductor POTRA algorithm ranked mRNA-mediated dysregulated pathways from the TCGA's project samples. The Neo4j graph database software was used to build, visualize, and analyze the resulting graph networks. And the analysis identified the most common and influential, highly ranked dysregulated pathways in 17 cancers. And this was from the TCGA open access dataset. So in study 2, dispersion analysis of POTRA ranked mRNA-mediated dysregulated pathways in breast invasive carcinoma from a TCGA pan-cancer study actually borrowed the pathway ranks from the first study. And so in the CGC, the POTRA ranked mRNA-mediated dysregulated pathways were studied using randomly sampled data sets with various solid tissue, normal primary tumor ratios and sizes drawn from the TCGA breast invasive carcinoma project. And the robustness of the POTRA algorithm to rank mRNA-mediated dysregulated pathways was also explored. For study 3, polytherapy EI, this is an interactive artificial intelligence platform for multi-omics combination drug treatment. And so we started a lot of our initial analyses and testing on the CGC, where we ran epitranscriptomic analyses to determine if ncRNAs are good predictors of cancer. And based on these results and other experiments, we will create a deep learning graph network algorithm and design a platform for it. So here are our main workflows for these 3 studies, just very high level. So the first workflow in this lighter blue, we began with the CGC's TCGA GRCH38 dataset. We filtered and collected all the mRNA samples, solid, normal, and tumor. And then we applied our prep tools and POTRA pipeline through our Docker Rabix applications. And then I built the graph network with Cypher, and I analyzed it with Neo4j. And the point here, the goal of this was to be able to detect the most influential mRNA-mediated dysregulated pathways in the pan-cancer graph network. And then for the second study, it's right here, this black workflow. So we kind of splinter off from applying the prep tools and POTRA pipeline, and we start with collecting the invasive breast cancer pathway ranks. And then we perform a dispersion analysis of these ranks. And the aim is to identify the combination of solid, normal, and tumor that produces the least deviation in the ranks. And then this final workflow here, we build the druggable mutations graph networks for each of these TCGA projects. And we built in our Shiny platform mockup, and we tested the ability to host interactive graph networks at different scales. And then we determined if ncRNAs have predictive power for different cancers. And then we used unsupervised learning to build and test multiple models. And as our project progresses, we'll be able to predict combination therapies. And right now we're working on getting the POTRA pipeline into the CGC public apps repository, and hopefully that will be done soon for all of you who are interested in using it. So here is a preview of the typical CGC project environment. I think this is a really, really great environment because you have such fine-grained control, right? For all of you who use HPC, if you like getting messages about your job starting, stopping, and erroring out, this platform can do the same, definitely. And you can adjust the settings so you don't have to receive any notification at all. You can invite different collaborators and CGC support team to kind of look through and help you through your tasks. So I just really love this because it makes it easy to reuse any pipelines that you build and quickly apply them to any data set that you choose. So this is the CGC app repository. So you have all sorts of different apps you can use in your project. I think it's really great, especially that we can actually see the workflows themselves and how they connect to different things. So now for the first study, pan-cancer exploration, mRNA-mediated dysregulated pathways in the cancer genomics cloud. So one of the most important steps of any study is really to be able to get your data set as accurate as possible, right? And I think this TCGA data browser that the CGC created is really excellent in helping you hone that data set that you're really looking for and understand it better. You know, you can also stratify by treatment, I think pack years for smokers, age, all sorts of things. So it's a really, really great tool, really beats the original TCGA method of finding these samples, right? And so yeah, this is, in this workflow, for all of you who are new to this, it allows you to import primary tumor and solid tissue normal samples at the same time. And you don't have to worry about mixing one up with the other because you actually can see all of these different attributes within your project folder, and you can actually filter by any of these attributes, and so you can't really go wrong. And you can also label your individual files. So now on to the POTRA tool. So POTRA stands for Pathways of Topological Rank Analysis. POTRA measures the relative topological ranks of genes in each biological pathway, then selects hub genes for each pathway and uses Fisher's exact or Kolmogorov-Smirnov test to determine if the number of hub genes in each pathway is altered from normal to cancer. And the value of detecting dysregulated pathways, well, recent studies have shown that both dysregulated or frequently mutated pathways should be used for the characterization of cancers instead of driver mutations. And network biology is an important approach that can detect frequently dysregulated pathways in distinct cancer types. So here's our workflow. So the POTRA algorithm was applied to 17 open access TCGA project data sets of FPKM normalized mRNA genes from primary tumor and solid tissue normal samples in the CGC cloud. And then Docker and Rabix were utilized for wrapping these libraries, computational tools, and pipeline building. Neo4j cipher language was used to visualize and analyze the ranked dysregulated pathways. So you can see here our workflow. I think what a lot of people like to do traditionally is to just they group up all of their normal samples and their disease samples, and then they quickly run a statistical test, right? But there's a lot of power to using repeated random resampling, where you're literally taking this unbalanced data set and then actually creating several subsets that are balanced, right? So for instance, if you have 100 normals and 1,000 cancer samples, right? So you can actually take those 100 normal samples and compare them to randomly resampled cancer samples, right? So it's like a 100 versus 100 over and over and over again until all of the different cancer samples have been represented without repeat. And then over here, we have the normal and cancer gene expression networks that POTRA builds. And then there's a detection of pathways with altered number of hub genes. And then this is followed up with an aggregation of metrics. So what I really like about the POTRA algorithm is that it's not just, taking the traditional average of these p-values, but it's using the log sum. And that's a much better method for taking average of these p-values. And then, of course, we use them to rank the final list of dysregulated KEGG pathways. But that was just for this study. POTRA can actually work with multiple different types of pathways, like Reactome is one, PharmGKB, SMDP, wiki pathways, etc. So here are our Rabix pipelines for the POTRA analysis. Up here we have we have on this side just two separate workflows for normal samples and tumor samples. And then we feed those results into this workflow over here. And we're actually performing an inner join. So we're actually creating a table of where we have columns of normal and we have columns of tumor. And then we have, then we go on to the POTRA correlation network algorithm, and then POTRA aggregation, we're just taking all of the metrics and we're just outputting the different ranks for all of the dysregulated pathways, along with some other metadata. So for all of you who are not familiar with Rabix, your workflow typically will have nodes that are green colored, like this one. When these nodes become orange, it's because this particular application has been updated. And so in order to make this green, all you have to do is click on it and then click, there's a side panel that will show up, and you click update and then it will become green. So also one of the great advantages of Rabix is that it allows you to keep copies, like limitless copies of different versions of your workflow. So in case you mess up, you can actually go back several versions and just apply that workflow to your data without a problem. So here are some of our results over here. As all of you know, TCGA has cancer projects with several different subtypes. And so since we were looking at dysregulated pathways, I really thought it would be important to understand how different normal and tumor are, right? Because in some instances, sometimes the normal tissue is sampled a little bit too closely to the tumor. And so we end up getting expression values that overlap, right? And so we see only minimal overlap, and some of these, like Luad and Lusk, have some minimal overlap. This one was a better separation, but still we can see where this is ACG tumor and PCPG normal. They start to overlap a little bit over here. But otherwise, I think we had really good separation for these subtypes. And here we have these subnets that I created in Neo4j. And so basically the edges are labeled by the bins that these dysregulated pathways were sorted into. So for instance, we have top 10 dysregulated pathways, top 20, top 30, 40, all the way down to 60. So pathways in cancer actually ended up being the one that was the most dysregulated pathway in these particular cancer projects right here. And then we have the next one, MAPK signaling pathway. It has the next most top 10 ranking, and then so on here and so forth, from RAS signaling pathway, and this one, and then exon guidance. You can see how they differ, like the colors and the arrows are different widths and colors, just to show you the bin that they belong to. So here are our final tabulated results for the different cancer projects. I think it's really interesting that for some of these projects, the highest rank can be quite low, like 43, we have 82, and then 50. And I think that this also signals that an ensemble approach might be really great, right? So if we combined our KEGG ranks with Reactome ranks, and wiki pathway ranks, and we can perhaps see this these rank values go higher. So, but this is I think this is a really great start. We have here results for signaling pathways, metabolic pathways. And I think this is a really great, quick way to find out which dysregulated pathways to focus on for particular cancer types. So here's some more results. So this large figure here is the big graph network that represents all of our results. Excuse me, all of our results. The purple nodes are the cancer projects, and the gray nodes are all of the dysregulated pathways. And here, Neo4j is a really great tool, allows us to color the edges depending on what we want. So for here, blue is top 10 most dysregulated pathways, and so on and so forth. And then Neo4j also allows us to run a PageRank analysis, so we can see the most influential dysregulated pathway. Here it's MAPK signaling pathway, and yeah, so the PageRank analysis determines the most important dysregulated pathway by traversing the graph network and checking with which pathway node is connected to the most highly ranked pathways. So for that, I really like Neo4j. I just wanted also to touch upon CGC App Preparation. The CGC is really great in giving users guidance for how to prepare these. And of course, before my lessons here that I can share with all of you is that before you start this, I would really recommend going into the CGC public app gallery and studying the different workflows, how they connect, and so you can see the possibilities as well as the limitations. And also each CGC app requires a development, pre-build, and commit project folder. All of them are separate. And yeah, the workflows that will become CGC apps can be tested in the CGC platform's workflow environment. And this is better than the Rabix desktop environment because once you're in the CGC, you can actually access data sets to test all your tools with. And over here, this very large panel. So Rabix, for each app that you make, you have to have a Docker container. And that's where you actually put in all your different libraries, your packages, and then you can put whatever else you like in there. So for me, I really like using Microsoft Azure, and I thought this is, a really, really simple way to create a great virtual machine for creating Docker containers, and then transferring them to Docker Hub, doing your pushing, pulling, and yeah, so I just wanted to share that with everybody because some other clouds like AWS, they can have a little bit more involved process with setting up virtual machines. I just thought this was super simple, and so I decided to share it. So our final CGC app for POTRA will look something like this. So we have all of the convenience steps. So basically, all you'd have to do is decompress your data, and then you feed the data sets into this workflow, and then your final output are the ranked results for the dysregulated pathways. And I will make it possible so that everybody can actually specify the type of pathway database that they like. So no one's going to be limited to KEGG. So conclusion discussion, overall, the POTRA tool can accurately identify dysregulated pathways, even in tumor samples with intratumoral heterogeneity by using a majority rules approach. And also the POTRA algorithm can identify dysregulated pathways by using only mRNA, while other algorithms arrive at similar conclusions by using a multi-omics approach. The most consistent ranks were found for the following primary sites, in case anybody was curious. It's breast, colorectal, kidney, liver, lung, prostate, and thyroid. And the POTRA algorithm accurately identified the dysregulated pathways associated with each TCGA cancer project, and it's also easily used in the cancer genomics cloud and therefore suited for large-scale analyses because it's also multi-threaded. I mean forks. You can specify multiple forks. And the POTRA algorithm will continue to be developed by the Dino Lab at ASU's College of Health Solutions. So overall, the CGC's TCGA data browser is an excellent tool for properly stratifying the existing data sets and helps researchers accurately select only the most important data sets and data types. The CGC task environment is critical for tracking down previous analyses and previous results files, as well as methodologies that were utilized to perform the analysis. And Rabix is a powerful tool that can help speed up bioinformatics analyses by enabling researchers to quickly select large amounts of data as input for a single algorithm or complex pipeline. And the CGC bioinformatics staff are effective partners in troubleshooting unexpected errors. And the CGC platform makes it easy to run very large-scale bioinformatics analyses of your choice. Now on to the second study, dispersion analysis of POTRA-ranked mRNA-mediated dysregulated pathways in breast invasive cancer from a TCGA pan-cancer study. So here we return to our data browser just to emphasize this point. It's a really great tool to use to find your perfect data set and keep stratifying as much as you like. Some background in data mining and machine learning, class imbalance impacts the accuracy and error rate of classifiers. And similarly, in bioinformatics, computational tools that are applied to unbalanced data sets will have more variation in its results. So the ideal data set ratio should be 1:1. Here are some of our earlier results from the last study. So just kind of highlighting that we're using the breast cancer project. And this is why, because they have the most normal samples, the best combination of normal samples and tumor samples, because the tumor samples, I think it's the most for these open access projects. And here's just another PCA where there's some limited amount of overlap between normal and tumor. So here's a dispersion analysis workflow in the CGC. So to identify the optimal solid tissue normal primary tumor sample ratios, the standard deviation analysis used random combinations of 1 to n unbalanced solid tissue to normal to primary tumor data sets with 1 to n, where n is 1 to 9. And to identify the minimum sample size, randomer sampling of solid tissue normal primary tumor samples, various sizes were used, 3 to 3, 5 to 5. This is basically testing the 1-to-1 ratio hypothesis, right? And going all the way down to 113 versus 113. Here are our workflows. And here are the results we got. So the average standard deviation of non-permuted ranks is represented here. So we see that the average standard deviation starts to level out at sample size 50. And then over here, we see that the average standard deviation is the least, it's the smallest when the ratio is 1 to 1. And then here, when we look over here, we're seeing permuted average ranks by ratio. So it's average rank versus ratio. And we can see here that ratio 1 to 1 has the most, it's the least varying ranks. So these results suggest that 1-to-1 ratio, solid tissue normal and primary tumor, achieves the lowest average rank variation, that the minimum sample size of 50 normal and 50 tumor sample reaches a steady state in the average rank variation. So in conclusion, future applications of the POTRA algorithm to analyze gene expression data sets such as TCGA should definitely use balanced data sets as well as a minimum sample size of 50 for both solid tissue normal and primary tumor to ensure the most robust performance. The present work also demonstrates how pathway ranks are changed by data set size. Interestingly, the MAPK pathway had the least variation in the different ratios of solid tissue normal primary tumor, perhaps because this pathway is very active in breast invasive carcinoma. In contrast, the CAMP 199 signaling pathway had the greatest variability because, perhaps this pathway is associated with tumor progression and therefore targeted by chemotherapies that are prescribed to the BRCA 201 patients. And in fact, the pathways with the greatest variability being CAMP signaling, human papillomavirus infection, PI3K ACT signaling pathway, and proteoglycans in cancer also had no detectable differences. and hub genes between solid tissue normal and primary tumor mRNA networks in ratios 1/7 and 1/9. This may indicate that POTRA can potentially be used to measure the efficacy of chemotherapy that targets genes in particular pathways. And lessons learned. So in the CGC project environment, it is easy to reuse data files that were generated in previous projects. It's really great that in the task environment, it allows you to literally track down these files without having to go through each and every subfolder. And it's also easy to reuse previous tools that were created in Rabix, because literally you can go to one project and copy your application into your current project. So it's really that easy. And the CGC task environment also makes it easy to generate and collect images that require a lot of RAM. And the tag feature makes it easy to quickly label and relabel results files before they get lost in your project folder. And the CGC safely stores all the algorithms and pipelines that were built in Rabix for every project. And the CGC bioinformatics staff are, again, really effective partners in troubleshooting unexpected errors. So sometimes people do not, I think one of the biggest sources of errors would be from setting up your Docker file incorrectly. Maybe you didn't actually install your libraries correctly, or maybe something is deprecated, and so they can actually have that type of view into the type of errors that you might not have otherwise. So now to polytherapy AI. This is an exciting brand new study. And the principal investigators are Dr. Valentin Dinu, Associate Professor of Biomedical Informatics at the College of Health Solutions at Arizona State University, and Dr. Marinka Zitnik, Assistant Professor of Biomedical Informatics at Harvard Medical School. And the polytherapy AI project was born out of a need for an increase in alternative combination therapies and shorter run times for massive NGS analyses. I think all of you know what I'm talking about, right? Sometimes even aligning whole genome sequencing can take a week, can take a month, depending, or even years, depending on how large your data set is. And plus an easier way to quickly, accurately build and derive insight from massive scale heterogeneous networks, which are known as HETnets. Alternative combination therapies are critical for metastatic cancer patients, as all of you know, and especially during drug shortages where humans and animals are both impacted. And here quickly are some personal experiences with these cases. Last year, my great aunt in Peru was diagnosed with a very aggressive form of metastatic ovarian cancer and underwent chemotherapy. She tried different drugs, and after multiple blood transfusions and multiple strokes, she developed the typical brain and bone cancers and finally died. And also, my dog was taking a polytherapy, and one of his drugs, known as vetmedin, became unavailable because of a drug shortage. So I called his cardiologist, and she told me that there were no alternatives available. I mean, you can imagine any parent with a child going through the same scenario, and I think that's why it's really critical to have alternative drugs available through different combination therapies. So these real-world experiences create a very strong motivation in me to create a solution for qualified researchers that can help them decrease the mortality rate in ovarian cancer and increase alternative combination therapies for all diseases. So based on this, I reached out to my former lab supervisor, Dr. Valentin Dinu, and told him about the platform, this concept I had designed for predicting combination therapies, and we developed it some more, and then recruited some of the finest bioinformatics researchers from Mayo Clinic, Harvard Medical School, and many more to continue developing the polytherapy AI project. And then we also recruited Microsoft Research. So petabytes of data, complex bioinformatics algorithms, and an international crisis known as the COVID-19 pandemic highlighted the need for a gold standard platform capable of accelerating massive scale analyses of heterogeneous data sets, multi-omics, clinical, etc. And the polytherapy AI platform project was created by me and my team so that the Microsoft Quantum Azure Cloud and quantum machine learning could make it possible to accelerate already trusted bioinformatics algorithms by the speed of light, especially for the prediction of multi-omics based combination therapies. therefore, we're excited to announce that for the first time ever through our project, qualified biomedical and bioinformatics researchers in the United States will now be able to leverage late-stage quantum technology, of course, with reduced error, to easily predict combination therapies and use the Microsoft Azure Quantum Cloud to quickly and easily accelerate any bioinformatics tool of your choosing, including custom code, which is, I think, a really great step for anybody who knows that it's such a kind of a progress killer when you're faced with a brand new computing language and no one in your lab has the time or ability to learn it, right? And so especially, I think, in times of crises like this, something like an acceleration envelope where you just stuff in your data and your program, you can just hit run and see it accelerate. That's the speed of light. Is going to be really critical, not just for now and probably also for the years to come. So with special attention to COVID-19, we will be creating, excuse me, we will be creating CRISPR-Cas9 drug screens using organoid disease models. COVID-19 is going to be included in one of them, but we will also be including several other non-cancerous disease models, but also metastatic ovarian cancer. So these are some microscopy images of organoids for different organ types. And so the HoloLens. So some of you might be familiar with these headsets. VR is really popular these days. So I think one of the greatest challenges of driving insight from humongous amounts of data, both clinical and genomic, is trying to fit everything on your desktop monitor and then trying to quickly take the gist of what you have, right? All your results. And so I really want to use this HoloLens to create a very rich, immersive, very cool environment where you're not going to be limited, right, to how many windows you can open. You can see all of the insight that was derived from the clinical informatics analysis, the genomic analysis, because everybody has multi-omics data sets. When you run all your data through a pipeline, you don't just want to see the results, but you sometimes want to see the intermediate results, right? If possible. So that will give everybody the ability to kind of step into a really cool environment and just look at everything all at once, and not just stop there, not just stop interacting, but also be able to perform analyses and subset things, especially with heterogeneous graph networks. I think that's also really critical because they're so demanding with memory, with RAM, right? And so, yeah, it's going to be a really amazing step forward for augmented reality and virtual reality, especially in the biomedical industry. And I think it's something probably long overdue. So with that, so one of the great things about polytherapy AI is that it's going to really support explainable AI. Explainable AI, everybody wants to know what's inside the black box of these different deep learning algorithms, right? And so since polytherapy AI is a deep learning graph network algorithm, we're going to allow our users to query it through Microsoft Teams, through email, Slack, chatbot, text message, and then you can ask your project whatever you like to better understand your results, and it can shoot you back a subnet or other ways, visual ways or textual ways of understanding how polytherapy AI arrived at this conclusion. And you can also ask it other questions as well to understand the algorithm. So for polytherapy AI, I use the Cancer Genomics Cloud for our initial stage of development, kind of try to see what's possible before we really move forward. And so we started with pan-cancer druggable mutation graph networks for all of the open access TCGA data sets. And then I built the RShiny platform mockup to test the ability to host interactive graph networks at different scales with the CGC team. And then I move forward to determining if ncRNAs have predictive power in cancer. Indeed they do. And then I use that unsupervised learning to build and test multiple models. So here is kind of a glimpse at what this initial testing environment looked like. You can see how dense these graph networks get, and then different ways to visualize drop-down menus, clickable, interactive. And so my first step was to create a TCGA GRCH38 drug gene interactions interactive and searchable environment for each open access TCGA product using RShiny, which is what you see here. And then I drafted a polytherapy AI dashboard mockup in RShiny. The same thing as you see here. This has like little drop-down menus. You can go to different sections and see text, and the panels are organized differently to show gene expression, results, etc. And then I stress tested the RShiny for large graph networks with the CGC staff. And then, so this is where I kind of move forward into the ncRNA to see if they can actually be predictive biomarkers for cancer. And I found out that only a subset of these ncRNAs can be used as biomarkers for each cancer type. And then I validated our results with a biomedical literature search. And then I trained multiple deep learning models. And what you see here are the heterogeneous graph networks. They can be extremely dense, and there are a variety of tools that you can use to visualize these things, but the larger the data set, the longer it takes to separate different neighborhoods, color them. It can be, unless you have humongous amounts of RAM available, it can be extremely time intensive. So onto our pre-training. So for pre-training, we use unsupervised learning. Here you can see off to the right, our training loss and validation loss was extremely low. That was really impressive to me, actually, because I know a lot of people have a rule of thumb, stop at epic 200. You know, that's actually early stopping for the training. And so the fact that actually I was able to get an accuracy of 99.5 at epic 5 was amazing, and even 97.5 was really great at epic 1, you know, the first training. And that was actually our worst performing model. So that was pretty promising. And now into a lesson that's really important. People kind of, again, a lot of people are used to going into public data repositories, just grabbing everything, comparing everything, and then being done. But for sure in deep learning, imbalanced data sets will greatly negatively impact your accuracy. So here we have like a semi-balanced data set, and our pre-training accuracy is 0.98, which is really, really great. But as soon as we use this imbalanced data set, which some people might actually consider severely imbalanced, we can see that our F1 score really deteriorates to 0.24. So you can see the impact is pretty huge. So there are a variety of ways to solution this imbalance problem. Some people use permutations to pad the imbalance groups, but there's a lot of algorithms out there that can help you choose different ways to balance these data. But again, it depends on the data that you have, what you're comfortable using, etc. So it's worth a Google. So lessons learned, the CGC's TCGA data browser, again, an excellent tool for properly stratifying existing data sets and helps researchers accurately select only the most important data sets and data types. The CGC task environment is critical for tracking down previous analyses and previous results files, as well as the methodologies that were utilized to perform the analyses. And again, Rabix is a powerful tool that can help speed up bioinformatics analyses by enabling researchers to quickly select large amounts of data as input for a single algorithm or complex pipeline. It's really great flexibility. And the CGC bioinformatics staff are effective partners in troubleshooting unexpected errors and for benchmarking. And the CGC platform makes it easy to run very large bioinformatics analyses. Some important points. You can reduce variation in POTRA results by using data sets with a minimum of 50 cases and controls. This is actually necessary if you're using the bioconductor version of POTRA. You have to make sure your input data sets are balanced before you go in. That's just to get the most robust performance. But if you use the CGC app version of POTRA, you actually don't have to worry about doing the random resampling and things like that. It will be done automatically for you. So that's a benefit of using the CGC. And then NCRNAs are indeed good predictors of cancer, and there are several studies out there that will back up that conclusion too. So future work beyond polytherapy would include expanding the POTRA pipeline available on Bioconductor right now to include multiple genomic data types and advanced visualizations. And of course, Dr. Dinu's lab will be heading that up. And I would like to thank the Cancer Genomics Cloud team for really being so helpful from the beginning, and the National Cancer Institute for the funding, and the Cancer Genome Atlas, and of course the Polytherapy AI team. If there's any questions, you can reach me, Margaret Linnen, or Dr. Valentin Dino.
Wonderful. Thanks so much, Margaret. That was a power-packed presentation. Very interesting to learn about the 3 different studies, and you tried connecting the studies, and also how it's going to be valuable for future work. So I'm going to announce to all the attendees, if you have any questions, please post in the chat box. I do have a couple of questions for you, Margaret. So you mentioned you did the, for your study 1, you used Neo4j, right? Was there a specific reason for using Neo4j, or have you compared with some other kinds of approaches and then chose Neo4j? Can you talk about that?
Sure. What I liked about Neo4j is that for people who, I guess, want something like an interactive environment where you can kind of point and click and then get what you like, it's really great for that. So for instance, someone might think, okay, so they might think, do I really want to learn how to code different labels into over 100 edges? Or do I want to kind of point and click and get that done like within 5 seconds? So that's what Neo4j is good for. And yeah, I think it's you can you just point and click on the different nodes. They give you also a variety of algorithms you can instantly apply to your data. And yeah, I think it's for people who don't have the time to kind of troubleshoot kind of more complicated code for manipulating their graph network. Neo4j is really convenient. And I think they're always adding new graph network algorithms to their library. So that's also an added plus, because it really eliminates any like troublesome errors and need for troubleshooting. Like anybody knows who downloaded our libraries or Python packages, sometimes the installation is a pain. Dependencies are there or they're not there, or you have the wrong version of Python or R, and it's just kind of a migraine, right? So yeah, so the fact that Neo4j has all of their algorithms and libraries immediately available is such a time saver too.
Great. Yeah, I understand. It's one main advantage is the simplicity of using that tool. Great. So we have a comment, amazing speech. So yeah, thanks again. One more question I had was, yeah, we all know, I mean, TCGA is such a powerful data set, and it's a wealth of information that we have. And every time I hear about some new studies coming from TCGA, it really, makes me wonder, like, there's so many possibilities. But I was wondering, have you tested this algorithm on any other data sets, or are there plans in the future to expand to other kinds of data sets, cancer studies? Yeah.
Cancer studies, I think sure. I think that the potential is there. I mean, definitely there is such a rich data set, the different data sets available through the CGC, and definitely they're worth exploring.
Great. And a related thing would be, how easy would it be to, modify or adjust the parameters for other kinds of data sets?
For POTRA in particular, that's one of the things that Dr. Valentino has been thinking about. He really wants to make it usable for all sorts of different genomics data types. And actually, I think we almost got there before the pandemic, but definitely it's a work in progress, and definitely that's a goal.
Okay, great. We have a question about the polytherapy AI computing platform. So what alternatives you had to Azure and why did you choose it? You already mentioned the ease of use. So are there any pros and cons you consider? And how about costs?
Okay, so so the reason we chose Azure was because, for instance, everybody has heard about quantum supremacy, right? So IBM, Google, and even Microsoft, they all have their different quantum technologies. And so Azure was the best because Microsoft was the best because they're actually working on greatly reducing the error that's impacting early stage quantum technology. And that error actually impacts the mathematical output. So that's why it's so important. I don't know what's going on at Amazon AWS. I just know that they have made some quick quantum technology available, but their qubits are much lower. So, and also I think with Google, I don't hear a lot about what's going on with their quantum, what their focus is. And with IBM, I think for sure their focus is more on the circuits, like circuits, quantum circuits and things like that. So it's not like immediate for general application, right? The way Azure. So Azure was the best suited for this task, and also were very generous and gave us access to their whole research department to support our project, and also give us a space on the quantum Azure cloud so that polytherapy AI can be sustainable, which is, as everybody knows, web tools. We all have our favorite web tools for genomics, but then they disappear, right? Because funding runs out. And so Azure is able to give us that sustainability cost-free. So that was a really great thing they offered. Pros and cons for Azure. Well, in general, Microsoft Azure is more, it targets the industry, Fortune 500 industry, where there's the budget is millions, billions. It's not really for life sciences. It's not priced for that, but there are special grant programs, right, for biomedical studies. They give you some amount of credits, and then you can start using their resources. Also, NIH strides finally including Microsoft Azure. So the prices are already going to become more affordable for biomedical researchers. Actually, what surprised me recently was when I was looking at different virtual machines, how the prices have really, really dropped for using their different virtual machines. I think in some ways, in some cases, it might be a little bit cheaper than AWS, Amazon. Yeah.
Okay. We have another question. Do you think the choice of normalization impacts POTRA analysis?
Normalization. Well, we use the FPKM normalized. I think impacted. Well, yes, well, probably slightly. I think in gene expression analyses, there's always some sort of normalization. People even do clustering, right? So that pre-processing step will impact your results, but probably not for the worse.
Okay, great. We have another question. Could you elaborate on the pathway dysregulation, how the pathway dysregulation is calculated, and could the method be applied at a single sample level?
Single sample, like I think single sample, yeah, I mean, as long as, for this is gene expression, right? So as long as you have like 150,000 genes, or at least minimum 80,000, I think, yeah, definitely you can do one cancer to one one cancer to one normal sample type of comparison. That's definitely possible. And so how pathway dysregulation is calculated. So let me go back to that slide. I think I had it here somewhere. So basically, POTRA uses the PageRank algorithm to find the most influential hubs for case and control, or cancer and normal. And then it compares these the expression levels of these hub genes either using Fisher's exact or Kolmogorov-Smirnov, and that produces the p-values. And then from there, we use the log sum to average these p-values across multiple samples. And then we sort on that, and then we start deciding what our ranks are, right? So rank 1 would be associated with the smallest p-value.
Okay, great. And one more question. When you show that you had good performance even at epoch 2, is it for training or test set? How did you make sure that the model was not overfitted?
So I basically, so you had, is it for training? So this is for pre-training, just to show as a part of a lessons learned and quick example. So this was pre-training. And so how did you make sure the model was not overfitted? So definitely you have to use a balanced data set, right? And so when you get a balanced data set, I think you have a good distribution of multiple groups. So it's not like one is dominating all the others, right? And so I think that's a good way to prevent overfitting. Definitely for the semi-balanced one, we made sure that the sample sizes were close enough, kind of just testing how much imbalance it can tolerate. So these are just kind of examples.
Great. So we don't have any more questions. So thanks again, Margaret, for the wonderful presentation. It was a pleasure to listen to all the different projects that you've already worked on and the exciting new projects that are coming up. I just want to share the screen and show information about the upcoming webinar in the next month. So let me just go forward 1 second. Okay, so next month in the CGC webinar series, we have Dr. Tatiana Popova, who is a senior scientist from the Institute Curie from Paris, France. So she is going to present on her work performed on the CGC to understand how splicing factors SF3B1 and SERPB1 and how the mutations in these splicing factors lead to aberrant acceptor usage. So stay tuned to the Cancer Genomics Cloud webinar page. You will see the registrations open up very soon. And once again, I would like to thank Margaret and Dr. Valentin Deno's team for the excellent work, and Margaret for presenting at the webinar today. Thanks everybody for joining. I hope you have a wonderful rest of your day. I will meet you all next month. Bye.
Bye.